נו, בסדר. אין לי מה. לא נמצא להדליק כזה. דיברתי איתכם גם בטוב. בבקשה. Okay. Hi everyone, thank you for coming. My name is Tal. I'm currently a postdoc at ETH Zurich, and today I will be presenting my first postdoc project, which deals with the extent of human-made pollution of anthropogenic zinc in the remote open ocean. And this is constrained by zinc isotopes in marine particles and aerosols. So first, to start off with a brief introduction about oceanic trace metals. So by trace metals, I mainly mean these elements right here. And trace metals and their isotopes in the ocean are excellent proxies for various ocean ocean processes at various interfaces. So the main ocean interfaces include the ocean and continent interface, the interface of the oceans with seafloor sediments, as well as the ocean crust or hydrothermal vents, and the interface of the ocean with the atmosphere. In addition, we can use trace metals and their isotopes in order to track ocean circulation patterns, both modern patterns as well as patterns of ocean circulation in the past. And many of these metals participate in biological activities in the ocean. So those metals that participate in biological activities are used as micronutrients. And these mainly include iron, cobalt, copper, zinc, cadmium, and nickel. So for those of you who are not familiar with these kind of graphs, these graphs in this case represent ocean transects. So these specific figures represent a transect of a ship going from the North Atlantic to the South Atlantic. So the x-axis in this case is latitude, the y-axis is depth, and the color represents concentrations in this case. So we can see that the upper ocean is depleted in micronutrients such as zinc and cadmium, as these micronutrients are actively assimilated into phytoplankton in the process of photosynthesis. And because these are used as nutrients, they are often correlated with macronutrients such as silicate and phosphate. However, at high concentrations, some metals can be toxic to marine life, and these metals mainly include copper, but to some extent also lead, mercury, and zinc as well. So let's briefly go over the main sources and sinks of marine trace metals, and we will focus on the ones that are important to this story. So we have rivers that contribute metals to the ocean at ocean boundaries. We have sediments and hydrothermal vents that contribute metals to the deep ocean. But these were also shown to actually act as a sink of metals. But the main source of metals to the remote open ocean is atmospheric dust. So dust that travels in the atmosphere can travel for very long distances and has the ability to reach remote oceanic environments. And as these dust particles settle on the surface ocean, they settle through the water column and they partially dissolve and release metals to surrounding water. And these metals then become available for marine phytoplankton. So because atmospheric dust is so mobile, it has the ability to reach even the most remote oceans. So in this case, this is, I'm sure you all saw this before. This is an image of dust carried from the Sahara Desert across the North Atlantic. And not only it fertilizes the North Atlantic waters, it also fertilizes the forests of South America or Central America. But dust doesn't just deliver nutrients and fertilizes the ocean. Dust or aerosols also has the ability to carry anthropogenic pollutants into the open ocean. And these pollutants are mainly lead, zinc, iron, cadmium as well. And when these metals are emitted to the atmosphere during the burning of fossil fuels, burning of coal, and other industrial processes, so once these metals are emitted into the atmosphere, they can interact with aerosols that are present in the atmosphere, and then get carried away in the same way dust is carried away to the remote ocean. And this is how anthropogenic metal pollutants are carried from industrial areas all the way to the remote open ocean. In terms of sinks, so I mentioned biological uptake, which removes metal from the upper ocean. And a very important source that I will mention during my talk is the process of adsorption or scavenging. So in this process, metals that are dissolved in seawater will become scavenged or adsorbed onto particles that are suspended in seawater. So this process of scavenging takes metals from the dissolved phase and puts them into particulate phase. So we would expect that metals that are affected by scavenging to be enriched in particles and depleted in the dissolved pool. So I mentioned this two pools of particles and dissolved. So metals are constantly exchanged between these two pools, the dissolved pool and the particulate pool. And we distinguish between three main phases in the particulate pool. So three main particle types. The first is biogenic, so basically cells of phytoplankton that are suspended in the ocean. The second type is orthogenic particles, and these mainly include iron, manganese, oxyhydroxides. Orthogenic particles basically form in the water column from chemical processes, so they retain the composition of seawater. And as these orthogenic particles form in the water column, they actively scavenge dissolved metals. So we say that manganese oxides are the most efficient scavengers of metals in the ocean. So as these particles form, they actively take up dissolved metals. This can be zinc, nickel, iron, and others. The last particle type are external particles, and in the remote open ocean, this almost exclusively includes atmospheric particles. So either lithogenic particles, so just mineral dust that arrives to the open ocean, or these particles that I mentioned before that are associated with anthropogenic emissions. So in each sample we take from the ocean, we can measure just how much we have of this sample. So we have a lot of zinc or we have less zinc, but we can also measure the isotope composition of different samples. So the isotope composition of different samples not only tells us how much we have, but it also gives us a fingerprint that is used to quantify different sources, sinks, and internal processes that each element goes through in the ocean. So here we're focusing on zinc. So zinc has 5 stable isotopes, and the isotope composition of zinc is defined by the ratio of zinc 66 to zinc 64 in our sample relative to the ratio in a standard, and it's expressed in this standard per mil notation. So when we discuss the isotope composition of metals, we refer to it as being isotopically light, so enriched in the light isotopes, and this will make the delta zinc value low, or isotopically heavy, which is enriched in the zinc 66 isotopes, and this would make delta zinc high. So in this example right here, this is a broad estimate of the known inputs and outputs and processes that control oceanic zinc. So delta zinc is at the bottom here. So we can say, for example, if we take the process of particle scavenging, so particle scavenging leaves a certain fingerprint on the isotope composition of zinc. So during scavenging, during adsorption onto particles, there will be preferential scavenging of heavy zinc. So heavy zinc isotopes will be preferentially adsorbed onto suspended particles. And this would make the dissolved pool isotopically light and the particulate pool isotopically heavy. So let's take another look at dissolved zinc isotopes distribution, both isotopes and concentration. This is an example from the transects from the North to South Pacific. So we see, as before, that dissolved zinc concentration are depleted in the upper ocean and enriched at depth. And we can explain the distribution of dissolved zinc concentration fairly well. In terms of zinc isotopes, so we have a pretty constant or pretty well-known isotope signature of the dissolved zinc isotope composition of the deep ocean at around 0.5 per mil, and this is pretty constant throughout the oceans. But in the upper ocean, the isotope composition of zinc is isotopically lighter, with delta zinc values that are typically below 0.5 per mil. So one would expect that these isotopically light signatures would reflect the biological uptake of zinc from the upper ocean. But Actually, studies have shown that biological uptake is associated with a very small isotope fractionation. And if anything, the biological uptake should take up light zinc and actually leave the dissolved pool heavy. So we refer to this isotope composition of the upper and mid-latitude ocean as being sort of anomalous because we can't really fully explain it. But the two main theories have been put forward to explain this anomalous isotope composition of the upper ocean. The first is removal of heavy zinc through scavenging. I mentioned this before. So scavenging preferentially removes heavy isotopes and should leave the dissolved pool light. And in recent years, researchers have come forward and have suggested that it is not a removal of heavy zinc, it's actually the addition of light zinc to the upper ocean through aerosols that are associated with anthropogenic emissions. And indeed, these aerosols, we measure them both in industrial areas and in the open ocean. And these are characterized by very low delta zinc values, which in theory are able to drive these light isotope composition of the upper ocean. And also these aerosols are very enriched in zinc relative to mineral dust, for example. So it's becoming increasingly clear that dissolved zinc isotopes alone can only provide us with a partial answer regarding this ongoing debate. So in my study, I take advantage of two relatively new approaches. The first is zinc isotopes in marine particles, and not just in dissolved, which is what most people have done so far. I'm also looking at aerosols. And the second approach is coupling to lead isotopes. So lead isotopes are a very established tracer of anthropogenic pollution, of anthropogenic inputs. And if we couple measurements to lead isotope, we might learn something about the anthropogenic source of zinc. In this talk, I will only discuss zinc isotopes to just really focus on one isotope system. So I mentioned the processes that affect dissolved zinc. So let's think for a second how these processes will be reflected in the particulate pool. Really quickly, so let's imagine a dissolved delta zinc pool of 0.4 per mil, which is pretty typical to the upper ocean. We said that biological particles, biogenic uptake is associated with a very small isotope fractionation towards light isotopes. So biogenic particles should be either similar or slightly lighter than the dissolved pool. On the other hand, we have scavenging. Scavenging takes heavy zinc and sticks it onto particles. So the fraction of zinc absorbed onto particles should be isotopically heavy. And this fractionation is quite large, actually. And we would expect particles that are dominated by scavenging to have a delta zinc value of between 0.7 and 1 per mil. Lastly, we have anthropogenic particles that are present in the water that come through aerosols. So as these anthropogenic particles sink through the water column, they carry very light isotope composition. They will partially dissolve and change the composition of seawater accordingly. But those aerosols that sink through the water column that didn't dissolve, they should, in theory, have a delta zinc value of either lower or similar to 0.4 per mil, and perhaps also anthropogenic lead signatures if the source of anthropogenic input is similar. So this framework allows us to define three main research questions that will guide us through this talk. So the first one is what are the relative controls of anthropogenic inputs relative to natural processes such as biological uptake, lithogenic inputs, and particle scavenging? The second one is how much of each of these sources contribute to this anomalous zinc isotope composition that we see in the upper ocean. And lastly, if we understand these two, we will try to quantify the extent of anthropogenic zinc pollution in the remote open ocean. So focusing on this third question, we're interested in the remote open ocean because if we will find evidence of anthropogenic pollution in the remote open ocean, this would really highlight the extent of the human footprint on the oceans. So this is what we did. We went to the most remote ocean we can, the South Pacific Ocean, which we see right here. So just to put this into perspective, in some regions of the South Pacific, such as the middle of it, the closest civilization is actually the astronauts in the International Space Station. So these astronauts are closer to the middle of the South Pacific than people living in New Zealand or South America or anywhere on these islands. So this really allows us to scrutinize the extent of human pollution. So samples were collected during the GeoTraces GP21 cruise that trekked from Chile to New Caledonia. And during this cruise, particles were collected from the upper water column. So in this talk, I will mainly focus on trends in the upper water column and not show these profiles, these transects that I showed you before, and mainly focus on trends in the upper water column, where we expect anthropogenic pollution to be. So just a quick presentation of the methods that we use. So tracer metals by name are found in trace amounts of seawater. So this makes them very susceptible to outside contamination. And to put this into perspective, we usually use the example of dissolved iron in the ocean. So the amount of iron that is dissolved in the ocean is equivalent to 1 paperclip dissolved in 10 Olympic swimming pools. So any outside source of iron, which is pretty much in everything that we use, can completely contaminate the sample. So when in the field and in the laboratory, we employ clean sampling and laboratory techniques. Obviously, we work in a clean lab. We sample seawater for dissolved fraction and also particles using these trace metal clean sampling bottles, GoFlow bottles. These are mounted on a trace metal clean rosette. And even the cable, which goes as far as 6,000 meters, even this is trace metal clean, and even the winch is clean. So we take this entire rosette and we put it in a clean chamber on top of the research vessel. And this is where we filter the seawater. So seawater are filtered through these filters, which their mesh size is 0.2 micrometers. So we define particles in seawater and dissolved phase samples. We say that they're operationally defined. So we define the particulate phase as being larger than 0.2 micrometers, and this will be collected on these filters. And we define dissolved as whatever comes through these filters. At the same time, we collect these particles, these dissolved phase or these seawater particles. We also collect airborne aerosols through aerosol collectors mounted on top of the research vessel. And so far, this has been pretty standard, and we know quite a lot about the concentrations of metals in particles and in the zone phase, and people have been measuring this for a while. What's new here that we were able to measure zinc and lead isotopes on very low concentrations of marine particles. And this enabled us to produce the first basin scale transect of zinc isotopes in particles. Not only this, in theory, if we're interested in other isotope systems, we can also, in the same column procedure that we do in the lab, we can also separate samples for an isotope analysis of other metals. We can do for the same sample, we can do lead, cadmium, nickel, copper, zinc, and iron. So we can do all of these 6 isotope systems, providing that we have enough material. We also separated these particles for labile and refractory infections, but I won't be discussing the differences between these as there are no differences basically. So I will treat the results as a whole. So moving on to the results, presented here is the isotope composition of zinc in marine particles from the upper South Pacific Ocean. So this is the upper South Pacific, and this is deeper waters. So the x-axis represents delta zinc. The scale is from -1 to 1. And we see that most particles sit between 0 and 0.2 per mil. This is presented again potential seawater density, and we use potential seawater density because we want to compare the isotope composition of marine particles to the isotope composition of dissolved zinc. So the isotope composition of dissolved zinc is represented by these blue triangles, and this is taken from either cruises in the South Pacific, which surface waters from the South Pacific are advected into the South Atlantic, into the South Atlantic gyre. And these data are also taken from other cruises in within the South Atlantic, the South Pacific gyre. So here, perhaps the most striking observation, and perhaps the most important one in the study, the most important thing is that the isotope composition of marine particles is either lighter or similar to the isotope composition of dissolved zinc. In addition, when we compare the isotope composition of particles to the isotope composition of aerosols, this is this orange bar here, we see an overlap between what we measure in aerosols and what we measure in particles in the ocean. And collectively, this tells us several things. First, it tells us that the marine particulate pool is not likely to be governed by scavenging. So in other words, scavenging is not likely to be an important process here, because if these particles would have been governed by scavenging, we would expect these particles to be here, to be isotopically heavier than seawater. And instead, we see that they're isotopically lighter. So this leaves, means that these particles are practically not governed by adsorption, and they're likely either biogenic or anthropogenic in origin. But since we see this overlap between marine particles and aerosols, these aerosols, again, they deliver this anthropogenic zinc from industrial areas. They're also very enriched in soluble zinc. So this already gives us the first indication that these marine particles are governed by atmospheric deposition of anthropogenic zinc. But in order to further constrain this, we tested a different tracer. And this is the other tracers we tested are elemental ratios. So we can measure all kinds of concentrations in marine particles, and we can use different elemental ratios to characterize different sources and processes. For example, a very popular one is comparing metal concentrations to phosphorus. And this can tell us about the biogenic origin of different particles. So for example, here, this x-axis represents zinc to phosphorus ratios in particles, and the y-axis represents the isotope composition of particles. So we have a pretty good idea of what should be the zinc to phosphorus ratios in natural phytoplankton or diatoms. And this is around between 5 and 10. So it's a pretty small range. So we see that, for example, when we look at samples from the Tasman Sea, okay, not from the South Pacific, but from a different region, from the Tasman Sea, these are these blue diamonds. We see that zinc to phosphorus ratios over there pretty matches what we would expect from natural particles that are governed by biological uptake. So this is right here. But in the South Pacific, which is these green circles again, zinc to phosphorus ratios are orders of magnitude higher than what we would expect naturally. So this gives us a first indication of that we have excess zinc relative to what we would expect naturally in phytoplankton. So now we need to ask ourselves, okay, we know that zinc is associated with biogenic particles, with biology, so where does this excess zinc comes from? So we can test other natural processes, other natural sources. So the next source we would consider is a lithogenic source of zinc. And in this case, we can use zinc to aluminum ratios. Aluminum is a major constituent of mineral dust, and dust is the exclusive source of particles or zinc or other metals to the remote open ocean. So we can use zinc to aluminum ratios in our samples and compare them to zinc to aluminum ratios in the upper continental crust, which represents the background value of natural lithogenic material. And again, we see that zinc to aluminum ratios in marine particles is many, many orders of magnitude higher than what we would expect naturally. So we can definitely say that lithogenic sources are a very negligible source of zinc to the South Pacific Ocean. Lastly, again, we see these excesses. Maybe these excesses come from zinc that is scavenged onto particles. But as we showed before, this observation here supports what we see for the isotopes. So here we use zinc to manganese ratios. So zinc, as I mentioned, is mainly scavenged onto manganese oxides. And we can use the zinc to manganese ratios of iron manganese crusts, nodules, and pelagic clays to assess what would be the natural value of the natural ratio of zinc adsorbed onto particles. And again, if we compare our samples in the South Pacific, these green circles again, to what we would expect if they would have been governed by scavenging onto manganese oxides, again, we see that that marine particles are again many orders of magnitude higher. So this already tells us that probably these three natural sources don't play a role in governing elemental ratios of zinc. And in order to put this in order to quantify this, we took these ratios and basically put them in a 3N member mixing model. And this looks like this. I won't go into the details, but this 3N member mixing model is also based on elemental ratios. So over here, we have manganese to zinc ratios in the y-axis and phosphorus to zinc ratios in the x-axis. And we define three main end members. We define the biogenic end member, which is characterized by high phosphorus and low zinc, or natural phosphorus to zinc ratios that we know. We have a manganese oxides end member, which is characterized by the manganese to zinc ratios of these pelagic clays and iron manganese nodules. And we define an anthropogenic end member, which is very high on zinc and low on manganese and phosphorus. So the output of this model is presented in this histogram. So this histogram shows us what is the fraction of each end member relative in a relative frequency. And what's most striking here is that biogenic and manganese oxides, the biogenic and manganese oxides end member rarely exceed 20%, so the contribution rarely exceeds 20%, while the anthropogenic end member is the contribution is somewhere between 80 and 100%. So all of these tracers point out that the marine particulate zinc pool is overwhelmingly dominated by anthropogenic zinc, with virtually no trace of natural processes. And this observation was quite surprising. We expected to find some sort of evidence of anthropogenic zinc, even in this remote ocean, but we didn't expect it to be overwhelmingly dominated by anthropogenic inputs. And this observation is accompanied by recent observations on other metals that are also affected by anthropogenic inputs. So we have the case of lead, which is very well known. So lead was heavily used as a gas additive up until the late 70s, but was still used in other countries until the early 2000s. And as a result of this lead use, we saw very high lead levels at the surface ocean, but these have been steadily decreasing ever since the ban on leaded gasoline. But still in the oceans today, lead is heavily controlled by anthropogenic inputs. But this is well known. Recently, we saw evidence of cadmium, anthropogenic cadmium in the oceans in the North Atlantic. This recent paper shows that there are certain controls of anthropogenic cadmium, which we didn't consider it to be anthropogenically perturbed. And this is also something we see in the Southern Ocean, and also in a paper we just submitted. We also see evidence of dissolved of anthropogenic cadmium also in the Southern Ocean, which is also pretty remote. And in recent years, we've been starting to see evidence of anthropogenic iron in the ocean. And this has been known for a few years now, but recently we saw that not only we see presence of anthropogenic iron, we already start to see an effect of this anthropogenic iron on the bloom dynamics of phytoplankton. And this is very important. So we start to see evidence of biologically essential trace metals that are carried to the oceans in excess, which is not natural. And these excess inputs from anthropogenic emissions could, in a way, shift this delicate nutrient balance and affect the way phytoplankton thrives or phytoplankton works, and affect phytoplankton communities. And at the end, it might affect the manner at which these phytoplanktons fix carbon and remove it from the atmosphere. So to sum up, we studied zinc and lead isotopes in marine particles and aerosols from the South Pacific Ocean. The main observation, I think, is that the isotope composition of zinc in marine particles is isotopically lighter than dissolved zinc, but similar to what we see in aerosols. And together with elemental ratios and 3N and mixing models, we conclude that that anthropogenic zinc delivered by atmospheric deposition dominates the marine particulate pool of zinc in the South Pacific relative to natural sources such as biogenic uptake, lithogenic inputs, and zinc associated with scavenging onto manganese oxides. And this suggests that atmospheric deposition of anthropogenic zinc could explain this anomalous zinc isotope composition that we see throughout the mid-latitude upper ocean. But I will say that it is possible that what we see here may be unique to oligotrophic regions of the ocean. So oligotrophic regions of the oceans are regions with very low primary productivity. So this very low primary productivity, this very low biology in the surface ocean might increase this anthropogenic signal. Or in other words, maybe in places where we have more biology, these anthropogenic signals will be masked or even buffered in a way by biological activities. So this is something we're looking into now. Now we're looking into the equatorial Pacific, which is much more productive, but has similar fluxes of anthropogenic zinc. And we're trying to test this further by basically doing the same in another transect in the equatorial Pacific. So collectively, this study and others that that recently came out really highlights the extent of the human footprint on the marine environment, and we have evidence now that this anthropogenic pollution reaches even the most remote of oceans. And with this, I will thank you.
You're not.
It's one of the most, it's very good case to study the anthropogenic input of zinc. Did anybody do it?
Where, sorry?
The Mediterranean, because it's so oligotrophic.
Yes, that's the main plan. That's the next plan. So after the equatorial, we already have samples from the Mediterranean sitting in my office. And not only the Mediterranean is really oligotrophic, it's also under very high anthropogenic stress. So it kind of represents both cases. It's under high anthropogenic stress, but it's also oligotrophic. So it's very interesting to test this hypothesis over there, and this is waiting to be done.
First of all, knowing they work in a clean lab, it seems like a huge work, huge effort to measure these low concentration. But I have a question, because you didn't quite, or I missed it. How do you know signature of the anthropogenic source of the isotopes?
So we have, there are not a lot of measurements of zinc in aerosols, but the one we do have are very, very light. So they're very, very negative delta zinc values. We also measure the aerosols during the same cruise, and we see that they match what we see in particles. And these aerosols that we measured during the cruise, they're both enriched in zinc, and they correlate very nicely with lead. I can show this maybe. So this is the zinc enrichment factor of aerosols, and this is the lead enrichment factors, and we see a very tight correlation between the two. So obviously this zinc is associated with the zinc in aerosols. Anthropogenic zinc in aerosols obviously is associated with lead. This gives us a first indication of the fact that this is anthropogenic. It's also isotopically light, so it's lighter than zinc, for example, in the upper continental crust, which is kind of background. And it's also associated with lead isotopes. So all of this points out to the fact that this particulate zinc comes from anthropogenic zinc from atmospheric deposition.
What would be the source? It's coming with the fuel?
So we can't really know which process governs this. It's maybe fossil fuels, maybe it's coal burning, but using lead isotopes, we were able to kind of suggest that these aerosols come from either South America or Australia. Most likely it's a mixture between them, because, for example, the lead used in South America, at least in recent years, comes basically from Australian ores. So it's all a mixture. But these aerosols, we suggest that they are a mixture of South American aerosols and aerosols coming from Australia. And lead actually is also slightly perturbed by emissions coming from China. So this also might be a small N number.
עוד שאלות? תודה רבה, טל. מתנה.
המשך למי שרוצה.